Hello everyone, so today I will be explaining about the chemical antisepsis for part 1. As you can see on the whiteboard, these are the groups of chemical agents that are used as antiseptics or disinfectants in our medical field today. So the agents include acids and alkalis, halogens, salts of heavy metals, antiseptics of phenol group, oxidizing agents, alcohols, dyes, surface acting agents, nitroimidazole derivatives, and the last one is derivatives of nitrofuran. So the first chemical agent that I will be talking today is about halogens. Basically, the mechanism of action of antimicrobial activity is the denaturation of proteins of protoplasm of microbial cells by reacting with amino group of proteins, which is displaced by the hydrogen and oxidation of sulfhydryl groups. And then the denatured proteins will lose its activity. Halogens include iodine and also chlorine. For example of iodine solution is Lugol solution and also povidone iodine solution. The indication is for disinfectant of surgical field, disinfection of wounds, and also for the surgeon's hand. The side effects may include burning sensation and also dermatitis. For chlorine, the example of solution is chlorhexidine. The indication is for disinfection of surgical area and also the surgeon's hands, tools, burn surfaces, and for septic processes. The next group of chemical agent of the chemical antisepsis is acid and alkalis. The first one is acids, for example, boric acid and sulfuric acid. The mechanism, it will shift the pH to acidic side and causes protein denaturation of microbial cell protoplasm. The indication, it will be used for washing and rinsing of mucous membrane of mouth, acute and chronic otitis, and also colitis. The second one is alkalis, for example, ammonia solution. The mechanism, it will cause inflammation and exudate to shift to alkaline side and also reduce inflammation. The indication, it is used for cleaning, washing of hands before surgery. The next one will be the heavy metal compounds, for example, silver nitrate, aqueous solution of silver, mefenid acetate, mercury chloride, and also zinc. The mechanism is blocking of sulfhydryl, carboxyl, amino groups of proteins, and also enzyme of microorganisms. The indication will be for disinfectant for skin or unbraided skin, also precipitation protein interface with essential metabolic activities of the microbial cells, for example, like silver nitrate. The next chemical agent is the antiseptics of the phenol group, for example, Savlon, hexachlorophane, cresols, thymol, and eugenol. The mechanism is they act to denature and coagulate proteins and are general protoplasmic poisons. The indications are 3 to 5% solution for disinfection of furniture, household, hospital linen, as well as patient's discharge. The side effects are readily absorbed through intact skin and mucous membranes, which can cause to intoxication. Moving on to the next one is the oxidizing agents, for example, hydrogen peroxide and also potassium permanganate. The mechanism include oxidizing agents decomposed with the releasement of molecular form of oxygen that is responsible for antimicrobial effect. The releasing of oxygen makes foam that cleans and deodorizes putrid wounds and also ulcers. The indication is rinsing of the mouth and throat, also for treatment of wounds that are infected with an aerobic microflora. The last chemical antisepsis agent in part 1 that I will talk about is the dyes. For example, ethacridine lactate, which is Rivenol, brilliant green, and also methylene blue. The mechanism is they are more active against the gram-positive bacteria. Also, the antimicrobial activity of this group falls in the protein environment. So what are the indications? The first one is for washing of fresh and infected wounds, cavities, bladder, and uterus. This is for the Rivenol. The second one is for the treatment of skin with snitches, pyoderma, blepharitis, and others. This is for the brilliant green. As you can see, these are the pictures of the Rivenol and also the brilliant green. So that's all for chemical antisepsis part 1. Thank you.